I am Dr. Saigita Achrekar, Chief Scientist of Dr.Dhaniram Baruah and his research team. We were and we are still working on number of incurable dreaded diseases to find out its root cause and cure. Now, we have studied number of dreaded incurable diseases such as coronary artery disease, diabetes, hypertension, SLE, moya, cancer, HIV, etc. Now, today we are going to focus on HIV AIDS and its cure, how our molecules we have isolated from edible medicinal plants are working on it, and how it's functioning I am going to explain you. Many viewers ask us how we are successful when Western countries are not getting the answer for this. The main obstacle they are facing is the mutation. What is mutation? Mutation means the structure of this virus is continuously changing, so they cannot target the exact structure of this virus, and that's why they are not getting any answer for this. Now, with our Baruah siRNAs, we have found out the complete algorithm of this virus, and that's why we can target the exact structure of this virus. We are not allowing the entry of this virus, and further replication is totally stopped, and that's why we are successfully treating HIV patients. Now, this is very important to note that after attacking the host cells, now what will happen next? Virus is going to replicate. Now, where they are going to inhabit it, they will amplify and clone these microRNAs, and where they are going to stay. Now, what we have decided to do, we have isolated plant biomolecules from edible medicinal plants of Himalayan regions of India. These are named as Baruah siRNAs, that is Baruah small interfering RNAs. These are exogenous double-stranded RNA molecules which are non-coding. Now, how they work, that we have to explain. Dr. Baruah says, silencing is the wrong terminology which may lead to recurrence of the disease. Now, how Dr. Baruah's siRNAs are working? They completely knocks down the detrimental microRNAs amplification and cloning. In addition to that, the post-transcriptional gene silencing is totally permanent. There will be no recurrence of the disease. That is the main heart and success of our HIV treatment. Now, in very simple way, I will explain to you how our molecules are working. Now, first it will stop the entry of HIV virus. How it helps? It modifies the receptors, like for example CCL5. CCL5, 32 nucleotide base pairs are removed, and it will become CCL5 delta 32, so that virus cannot recognize the entry or the door to enter the host cell, and the further entry of the virus is totally stopped. Further, it produces the combat genes, Baruah combat genes, which activates the antiviral defensive machinery in the host system, so that this virus is not allowed to enter, plus the replication is stopped. There is no further mutation of the virus. In addition to that, number of combat genes, like it enhances the apoptosis of the host cells which are invaded with the HIV virus, so that the virus is getting killed. Apoptosis means the cell destruction with natural killer cells, phagocytosis, and plus in addition to that, it activates T cell receptors. That is the main concept of our treatment, so that we are getting 100% successful results with our treatment. Now, we have produced number of books with genomic mapping, and these are the full proof evidences you are having with us. So, nobody can question us. We are challenging scientists of the whole world to say that we are wrong. Come forward to challenge us, accepting their challenges, and that is the success of story of our HIV patient, and they are leading totally normal life. They have forgotten that they were once upon a time they were HIV patients.